Welcome to the third keynote session. My name is Christoph Finkler and I have the honor to co-moderate this session together with Harold. We will hear about an update on the ICH activities on the revision of ICH Q2R1, analytical procedure validation, and the development of the new ICH Q14 guideline on analytical procedure development. I'm happy to introduce our keynote speaker, Tona Agasøster. Tona works at the Norwegian Medicines Agency as an assessor of quality dossiers for marketed application and clinical trials, mostly on synthetic substances and finished products. She's a pharmacist by education and has a PhD in development of automated HPLC methods for the determination of some antibacterial drugs in fish using dialysis for online sampling preparation. She started in a position at the Norwegian Medicines Agency in 1998 and has been an assessor of many centralized applications. She has been a member of the Quality Working Party since 1999 and the respective core team since its introduction in 2012. She is a Quality Working Party observer in the EDQM's Expert Crop 7 on antibiotics and is the Quality Working Party and European Commission representative in the ICH Expert Working Group on the Q2 and Q14 development. She's also involved in EMA drafting groups. With that, I hand over to Tona.
Good afternoon, everybody. It's very strange. I can't see you, but at least I can see somebody I know. I see Christoph Finkler, and we are together in this expert working group working with these two guidelines. So I will try to tell you the latest development on Q14, and I will also give some background information. I have to give a disclaimer. The opinions expressed here are my own and not necessarily those of other EU assessors or other regulatory representative in this expert working group. Because I haven't discussed in detail with other EU assessors the content of this presentation. And maybe Christoph will also find that I say something that he doesn't totally agree in, but we'll see. So, a little outline of this presentation. I will say something on background and objectives. I will talk a lot about analytical target profile, which the industry is very interested in. And I will talk about the survey and other input from assessors. I will talk about the Q2 revision. And I will focus also on elements of the enhanced approach for analytical procedure development. And I will get into changes to analytical procedures and also next steps in this work. So the background for Q2, this revision, is that this guideline was developed a long time ago. It came into force in 1995 and it covers mainly chromatographic techniques. It has been a successful and beneficial guideline over 2 decades. But new technologies are arising, for instance, biological tests, immunochemical tests, multivariate tests, and hyphenated tests, and of course, associated new validation methodology. And also this old guideline lacks guidance around newer technologies. It's leading to incomplete submission data and also additional requests from authorities. And the guidance may be insufficient to establish the suitability of such new techniques. So, some background for Q14. There is no ICH guideline on analytical procedure development currently, and only validation results are normally presented in the absence of development data, which is a drawback. And the regulatory communication is ineffective, especially for non-conventional methods. And currently, applicants do not have opportunity to present scientific basis to justify flexible regulatory approaches to post-approval analytical procedure changes. So based on this situation, we would like to include validation principles that cover analytical use of spectroscopic and spectrometry data, for instance, NIR, Raman, NMR, MS, and some of which often require multivariate statistical analysis. And we would like to provide a general framework for the principles of analytical procedure validation applicable to products mostly in the scope of Q6A and Q6B. So now the methods for biological products are in scope for this work. And objectives for the Q14 guideline. We'd like to harmonize the scientific approaches of analytical procedure development and provide the principles relating to the description of analytical procedure development process. And we would like to improve the communication between industry and regulators and facilitate more efficient, sound, scientific and risk-based approval, as well as post-approval change management of analytical procedures. We would like to see this as a possibility to present different enhanced approaches when developing analytical procedure, or to choose the traditional approach, the classical approach, or a combination of both. I think we would very much like to see a combination to encourage to do as much as possible. We acknowledge that the knowledge base for analytical procedure is often larger than what is usually presented in the method validation section. And we would like to give the possibility to present additional information and benefit from that. So Q2 and Q14, they exist in a landscape. They are intended to complement or many other ICH guidelines, such as Q8 to Q12, which are Q8, 9, 10, 11, and 12, and the ongoing ICH Q13 for continuous manufacturing. And we have, in this work, we have to consider interactions and possible contradictions, and we need to align on key elements and terminology. So we see here this landscape of these guidelines, which are most, things are happening for now. We know that the Q12 was just finalized, and we have the Q14 and Q2, as you see, and also Q13. So the Q12 gives approval procedures for analytical procedure changes. Q14 gives guidance on analytical procedure development and also risk assessment in association with that. And Q2 validation and also terminology and examples. And Q13, which is of course employing process analytical technology, which is related to this analytical techniques. And I will also like to mention that in our work between the EU assessors, we have set up a discussion group related to Q14 and Q2, where we discuss with experts from Q12 and Q13, and we also have some biological experts in that discussion group. So there are many expectations here. The Q12 enables harmonized change procedures, at the same time allowing innovation. Q14 is going to give to encourage increased scientific understanding and connect process and product and analytical procedure knowledge, and to give harmonized methodology to manage and categorize risks. And the Q2 revision is going to include present and future techniques, and at the same time keep the excellent elements of the previous version, because it's a guideline that a lot of people know well and likes very much. So we have to keep the good things at the same time as we are developing it. So this is a picture of this expert working group, which and you can see there's really now a lot of countries participating in the ICH work, a lot of people from Asia. And here at this meeting, we are in Singapore, and that's now 1 year ago. We have not been able to meet after this, unfortunately, as you know. So we hope that will soon be possible again. And you can see here we have really many participants, and from the EC Europe, it's myself and Rob Bream from the EMA who are representatives in this work. So I will start with the beginning, which is not ABC but ATP, we have the quality target product profile, which describes predefined targets for quality attributes, and this concerns the manufacturing process. And we have a kind of analog for this analytical target profile, but at the same time relying on the quality target product profile, which is and this ATP is a prospective summary of the objectives of the analytical procedure with anticipated performance criteria to ensure the results are appropriate for the intended purpose. There will be set performance criteria for the relevant performance characteristics, and the ATP will drive the choice of the analytical technique and also direct the development of the analytical procedure. Well, the exact definition of the ATP, as it's currently now, is a prospective summary of the performance characteristics of the analytical procedure with anticipated performance criteria to ensure the results are appropriate for the intended purpose. And here in this figure, you can see this nicely presented. On the top, we have the intended purpose. And then we have the performance, the target, the criteria. So the intended purpose would be, for instance, an identity test, purity, assay, or potency. It could be a critical quality attribute as, for instance, charge variance. And the performance target would be criteria for accuracy, precision, specificity, and so on. So this is many would say this should be independent of the ATP should be independent of the technology selected. But as I see it, we have to consider also available technology and their possibilities when designing the ATP. We can't see it totally in isolation. And the technology, you can then select the most appropriate one, or maybe you can see that there could be several possibilities. You can choose, for instance, size exclusion chromatography, and then you have to design parameters which are suitable for that technique. That's of course very, very related to the selected technique. We have seen a lot of ATP examples from industry, which have been developed by the FBR Analytical Lifecycle Working Group. A collection of examples was presented with an article with an article called Analytical Procedure Lifecycle Management, Current Status and Opportunities, in Pharmaceutical Technology Europe in December 2018. Then in that article, there was 3 examples. The first one was measurement of potency using a cell-based potency assay for a biological product. And the second example was measurement of high molecular weight species in a biological product. And the third one was a measurement of impurities in a small molecule drug product. And EU assessors, we commented on those examples early 2019. The FBI is developing examples all the time, and these examples, which I will show in this slide and next one, I got from Christoph late. They are from late 2019, I think. So this is an example where we have a development in ATP. You have in early stage development, this first slide, which is it's a charge heterogeneity for a monoclonal antibody. And as you see, you have their performance characteristics to the left: specificity, accuracy, precision, and range. And then you have performance criteria to the right. And you can see there that there are relatively few performance characteristics, and the criteria are relatively wide. And there are also some additional technology-dependent performance requirements. For instance, if you use HPLC technology, the robustness is also relevant. And I would say that's always relevant, but maybe this is described based on the technology selected. And here you have an ATP for the same issue in a late stage development. And you can see here there are more performance characteristics described in this ATP, and also significantly more narrow ranges for those. And also you have here the additional technology dependent performance requirements. Well, some comments to these examples. We think that these examples are improvements to the examples previously presented. And we note that there are additional technology-dependent performance requirements included. And but we note that even if wide criteria may be acceptable in some cases, it's very important to understand where variability comes from. Of course, this allows for more reliable data. We're starting now to develop examples of ATP to include in an annex of Q14. And I think this is really something which is essential to this guideline to show how things work in practice. So we are really now getting to the point in our work. And we plan now to develop examples for a binding assay. And some people would like to also have in mind cell-based assay at the same time, but I think that would be challenging, but we'll see if we can manage that. And also an example on protein content. And we have our biological experts involved in that work. A third example which is planned is on small molecules, on impurities in a small molecule drug product. We think that an ATP should be sufficiently specific regarding the intended purpose of the method and what to measure. And we note that for instance, for biological products, there are many commonly designed critical quality attributes which are actually composites of several related parameters or attributes, for instance, aggregates or total impurities. And if the ATP refers to a composite critical quality attribute, then methods used may not be mutually interchangeable. So that's something to really have in mind here, and not try to have too wide ATPs. And we also find that it's difficult to cover relevant criteria if trying to cover very different techniques in one ATP, such as, for instance, if you try to cover a binding assay and a cell-based assay in the same ATP, that could be really challenging. We plan to give some guidance on how to derive validation criteria. This has been a request from EU assessors, and it's also very relevant for the analytical target profile. But we see that many industry people, they are very reluctant to this. So, so we have not been able to be very specific so far, neither in the Q2 or the Q14. But we think now that these ATP examples, that could be the place to try to say something on this. So we will try to then show how to derive criteria. From an assessor point of view, I think they should not be too wide, and we should not forget that we would like to see robust methods, and this could be if we allow very wide criteria here, that it's sending kind of the wrong message. So anyway, we need robust methods. So also we have this annex in Q12. We have the chapter 8 and annex 2 in Q12 on pharmaceutical product lifecycle management, which is described the structured approach for analytical procedure changes. Then you need a validation protocol, and you need acceptance criteria, which I think should reflect current expectations for method performance, be justified scientifically, and should not be less stringent than those used for the validation of the current method. Let's see what we can agree. I think it's going to be difficult, but I think we need to go into this. We can't just avoid it because it's difficult. So we will, of course, it has to be a case-to-case decision, and we would not aim at giving very direct recommendation of what the criteria should be, although some assessors and others would like maybe an indication of that, but we would more like to say something on how to do it and show it in these examples at least. We had a survey in this expert working group some time ago. It was a technical survey questionnaire. It was where the objective was to collect feedback that will assist in the revision of this Q2 guideline, to develop it to be as inclusive as possible to cover the validation of analytical procedures for all types of medicinal products. And the deadline for this was early 2019. And there we got significant input from EU regulators via BVP members, which was considered very helpful. And this input from the EU regulators has been the basis for teleconferences on how to proceed with the comments. And then we are now implementing this input into mainly the Q2 guideline. some input from the survey. These biological assessors and other assessors, it may be controversial, but moving to a Q12 life cycle paradigm would mean that validation criteria need to be assessed. And this would be especially useful for biological assays with high variability. An indication of what is considered normal variability, for instance, for accuracy and precision, for certain types of assays would be helpful. So that's something we can consider, and maybe we can reflect in the examples we are now developing. And also total analytical error, to have a single number which indicates the overall reliability of a specific result might be useful. Could be a joint criterion for accuracy and precision. And that's exactly what we are now trying to include in Q2. So that there will be a section on that in the revised Q2. A little lost here now. So another comment was no flexibility should be allowed in the absence of supportive robustness studies. We really expect that if we are to consider flexibility, then at least as a minimum that it should be a robust method. And regarding trending and real-life performance, it's noted that the validation is a snapshot at a point in time, and assays should include an assay control for trending purposes. I think we are trying to accommodate that as well. And also a section on system suitability tests and how those should be chosen to ensure consistent method performance should be included, and to give justification for the SST proposed. And some other comments from the survey when it comes to chromatographic methods for impurity determination in biologicals. It's noted that this current Q2 guidance on impurity testing is not so easy to follow for biological product, since the impurities can't be isolated. And there is no absolute purity evaluation, but relative purity compared to a well-characterized standard, which is characterized by orthogonal methods to the method under validation. So this is going. We are now working on this, and we're also describing better what orthogonal methods means. And since results depend on the analytical method, absolute purity is difficult to determine, and the direct comparison with validated independent analytical procedure is therefore difficult. And peak purity testing is difficult, since different forms of the large molecule cannot be separated by chromatographic means. So we have now included better description of relevant aspects here in the updated text in Q2, and we have adjusted the wording not to exclude biological methods. So this is something about the progress of this Q2 revision. As I said, we have updated the text in the main guideline and made it more general. And the methodology has been modernized to include newer technologies. A few issues have been moved to Q14, for instance, a system suitability test and robustness. And this is going to be a huge annex. We have included examples for common techniques. And some of our colleagues in this expert working group have argued that we should not have too many, but he hasn't been heard. So I think I think currently the plan is to go on with all these examples. And it should also be noted that these are examples, and they are not prescriptive for exactly what to do, but they are examples. So we have example for separation techniques, such as chromatography, of course, which is what is currently included. And also it's planned to include CME, quantitative techniques for product-related substances and impurities, for instance, the charge, heterogeneity variants, and oligosaccharide profiling. We are having an example for dissolution plus UV or separation for measuring. It has been a difficult discussion there because the FDA, the US colleagues, they have a different tradition in how to do this. But I think we are not so direct there anymore. We have an example on elemental impurities by ICP, OES or ICP-MS. We have quantitative NMR. We have bioassays for potency by binding assay and by cell-based assay. And that has also been a big discussion because in the previous draft, the first draft of this example was there. It was tried to combine both binding assay and cell-based assay in one example, and it turned out to be difficult. So our biological colleagues from the BVP, they have now, a few of those have been involved here, and they are now developing separate examples for binding assay and cell-based assay. And that's well underway now. And there is also a plan to have an example for a bioassay quantitation of impurity, for instance, residual host cell protein. There is example for quantitative LC-MS and also for quantitative PCR. And it was my proposal also to include an example of particle size distribution assay based on a dynamic light scattering and diffraction. And we have an example for NIR assay. I was asked to say something about assessor experience, and I think I've already indicated a lot of around that, but I have also one slide more directly on this. And in my experience, we need robust methods, and we should avoid situations where results for instance, assay and degradation products are fluctuating, and it's difficult to reveal any trends. Because that's what we need when we look at stability data. We need to see if there are any trends. And also, when we receive applications for marketing authorization, very often we need to base such assessment on very few measurements. We have the initial starting point, and maybe we have, in some cases, only 6 months data, and in some cases, maybe 1 year's data. But it's not so many measuring points, and then we need a method that can really give the correct results and clearly show trends. And I have encountered many times that companies state, for instance, in answer to questions, that the fluctuating results are because of variability in the analytical method. So I think we should avoid that by investing a little more in the methods and making them sufficiently robust. And also we see some examples of a kind of tick box mentality. So we would really encourage development of more knowledge of the background for the analytical procedure and see how things are connected. So I will talk now a little about the elements of the enhanced approach. And in the enhanced approach, we have a more systematic way of developing knowledge of an analytical procedure, which starts with the risk management, which is risk assessment and other quality risk management elements. But in this context, maybe most risk assessment. And then we have the robustness and operable ranges. So we have enhanced development in a more systematic way to see which are the critical parameters of an analytical procedure. And we have also the analytical procedure control strategy, which is identification of critical parameters and knowledge of their impact. We have detailed instructions and clearly specifying parameters which need control, and control of any critical reagent or other consumable. And of course, the system suitability test, which is also used in the more classical approach. And one other element, of course, of the enhanced approach is the change management, which we here in this guideline would like to see in connection with the development work invested. There should be some kind of connection of how much development work you have invested and how this can be realized in the change management. And also we have the real-time release testing, which we touch upon in this guideline. So what is space and flexibility? I think the space for flexibility is based on what has been investigated. You can't claim flexibility if you haven't investigated the background. But of course, in some cases, you have also other knowledge in addition to the investigations you have carried out. And for full flexibility, you have also to study interaction effects by design of experiments. We note as assessors that we very seldom now see applications where design space is claimed. I have seen a few, but seems that the industry is not so interested in that, and they would like they do maybe a kind of development which is close to a design space, but not totally. They don't do all they need, but only almost what they need to claim a design space, and they would rather have a lot of proven acceptable ranges. The Q12 guideline says something about established conditions for analytical procedure. It says for the classical approach, when more limited development studies have been conducted, this may result in a narrow operating window to ensure method performance. In such cases, established conditions may be more extensive with fixed and/or tight conditions. On the other hand, enhanced understanding, if you do enhanced development, can lead to wider operating window that ensures method performance, where established conditions can be reduced and focused on method performance, for instance, ranges rather than set points. But more precisely, we think that enhanced development leads to a more appropriate set of established conditions, which in most cases will be less strict, but not always. We very often hear that the industry would like to change between different technologies, but then you should have in mind that enhanced development and enhanced knowledge is needed, and this is related to a specific analytical method. And that kind of knowledge is not a basis for change to another technique. you need relevant development for the relevant analytical procedure. And that should also be reflected in the dossier. If you would like to change to a different technique, then you can use the mechanism of post-approval change management protocols, which is now well described in Q12. So how is post-approval flexibility interpreted? It could be from making small changes to a method without prior approval. Somebody would maybe like to change any aspect of a registered method without prior approval, or maybe change to a different method without prior approval. And maybe not so many would like to do that, but that's not possible. So Q12 describes changes to analytical procedure. As I mentioned already, when it comes to need to have some guidance on setting criteria, you have the chapter 8 in combination with Annex 2, which describes the structured approach to analytical procedure changes. Then the high-level description of the original and the revised method should be the same. And for instance, it could be within reversed phase chromatography with UV detection. This approach is applicable to various variations to analytical methods, and it's not linked to the development approach. And out of scope here is the test method based on biological and biological or immunological or immunochemical principle, or a method using a biological reagent. For instance, bioassay, binding assay, ELISA, testing for viral and adventitious agents. And also now in Q12, they have developed annexes. And one of those are on identification of established conditions for an analytical procedure. That's a relatively simple procedure. I think it was based on the pharmacopoeia method and for a biological product. And also there are examples for post-approval change management protocols. When it comes to guidance on changes in Q14, we try to build on what is already there in Q12, and we focus on risk associated with changes. We have risk factors, which includes relevance of test, complexity of technology, and extent of change. And we have risk reduction based on knowledge. It could be product and process knowledge, analytical procedure knowledge, and analytical procedure control strategy. And it also based on bridging by use of standard material or to previous methods. And here we can, of course, rely on, we can use the tools of post-approval change management protocols or other elements described in Q12, such as a product life cycle management document. And this slide describes almost the same. As I said, we have to the left, we have the risks, we have the risk reductions, the things you do for risk reduction, and also the pre-agreed, for instance, post-approval change management protocol, which altogether could give a reduced risk for a change. Lately, we have been discussing in the Q14 expert working group and also the Q2 expert working group, which is the same, the use of ATP as an anchor or foundation in change management. But I would like to point out that I think that what I tried to describe in the last 2 slides, I think that that's the most important way to deal with changes in this guideline. But also ATP, the use of ATP could be to some extent a tool here. We as assessors, we feel that the ATP should mainly be set up for development purposes. While because if companies develop ATP for with flexibility in mind regarding change management, they tend to be mainly too general and also less relevant, and also not gives, they don't then give so clear regulatory communication. And that's certainly an aim of this work, to facilitate good regulatory communication. So we think that an ATP could be used as a foundation or anchor regarding expected method performance. We then need a well-developed ATP based on knowledge of the analytical problem and the measurement. And as many would say, it's intended to be independent of technology chosen, but I would rather say the ATP drives the choice of the analytical technique. And this ATP could allow some additional flexibility dependent on a well-developed ATP. Then some changes to the analytical procedure could have a lower reporting category than they would normally have based on the ATP. And I would say especially in combination with other elements of the enhanced approach. In practice, the use of ATP within a certain class of measurement technology or analytical procedure based on similar measurement principle may be the most straightforward. And in line with Q12, changes to the method principles should be applied for, of course. And in general, the Q14 guideline has to be based on Q12. We can't do more than than is described in the framework of Q12. And when discussing with other EU assessors, we don't expect. We think you shouldn't expect too much regarding ATP as a tool for flexibility, and we think it's particularly useful for small changes, which I think is very frequent, and it should anyway. Then it could be a really useful tool. And when we now work with this work on these ATP examples. We also need to reflect on which additional knowledge we expect in order to be basis for some flexibility here, which is then mainly knowledge of the analytical problem and measurement. So regarding flexibility and robustness, I think clearly the aim here should be to reduce the burden of variations where possible. And Q14 should give guidance in presenting documentation that could be the basis for allowing flexibility. And the document, the knowledge and understanding would also demonstrate robustness of methods, and the guideline could act as a stimulus to develop robust methods. And I think we need to have in mind how to encourage the development of robust methods, and also in situations where full quality by design based on design of experiments is not developed. I think we should aim at facilitating reasonable flexibility supported by appropriate data. So I'm now getting close to the end of this presentation. I show here the work plan for this work on these guidelines, the expected future key milestones. And we are now having another meeting we have a week of teleconferences ahead of us soon. And in February, after that meeting, in February 2021, we see a round of intra-party consultation. So that second round of intra-party consultation will then mainly take place in January or and February. So we will have a long period there. So there will be good opportunities for everybody to consider these more advanced drafts to comment on them. And then we will review those comments, and we hope to have a step 1 sign-off in May 2021. And we really hope there will be a physical meeting, because then it's much easier to progress it. And I think we have this timelines for this work has really been stretched out. And of course, that could be because it's very complicated. But I think also a very important reason is that we didn't have the face-to-face meeting in May, and we won't have it now in November either. So the teleconferences are not so efficient, and there's in reality only a small time slot every day that is possible for everybody to attend. When we have people from California, the East Coast, we have people from Europe and Japan. So it's not easy in practice to have good teleconference to progress this efficiently. So next steps would be that this expert working group continues to draft and edit these sections in order to have draft documents for line-by-line review at this virtual Athens meeting coming up now very soon. It's in fact the week after next week. And we have now started also work on the annexes in parallel. So we'll soon have another important meeting in this project. So I think this is the end of my presentation, and thank you all for listening. Hand over to Christoph.
Yeah, thanks.
So I guess we can start into our Q&A session. Christoph, you wanted to start off with the question?
Yeah, so maybe I have a question, maybe also a comment to Tuna, not sure how it ends.
I don't hear you so well.
Can you hear me?
I hear you a little weakly, but I can hear you.
Okay, I go closer to the mic. Maybe it's better now. So I have a question to you related to the definition of the ATP. I think when you started around elaborating on the definition of the ATP, was described like the performance requirements for the analytical procedure. I better liked actually your description you had on the later slide, where you said it describes based on the requirements on the measurement. Could you actually elaborate on what it would actually should entail?
Well, I didn't quite get what you. I think the ATP needs to be sufficiently specific, and that comes to, for instance, the intended purpose. For instance, we have examples for high molecular weight species, and if that's not specific, what you are looking for, it could be in many cases for monoclonal antibodies. I understand from my colleagues that in practice maybe you only need to measure the dimers, and that's what, if you choose the size exclusion chromatography, that's what you're doing. So and if you was going to say something very general, it could it's not a good description of what you need to do, and it's difficult to set meaningful criteria for the test applied, because as I recall, the alternative method would be an UV-based method, and that's a totally different method. It wouldn't give so much meaning.
Okay, thanks for that. Another question is, when actually do you see one should develop an ATP, and how is actually the role of the ATP in the life cycle of the method?
Well, when I discussed this with my European colleagues, we see the ATP as most useful in the development, for instance, as an analog to the quality target product profile, to be an aim. so we think it's the most important function is in the development of the method, and of course you could also to some, and also I think, as I mentioned, it's a kind of interaction. You can't you don't see this in total isolation. You have to reflect on which techniques are available, and also what is the state of the art. You can't do this independent of knowledge of what is state of the art. And but if you invest a lot in this ATP and it's sufficiently specific, then it can also have a role in the change management, as I said, especially for small changes. And that could be really useful, I think, around the world, not only in Europe. That could facilitate things a lot. And also there are some possibilities, I think, with changes to relate the techniques that are closely related to the technique you already are using, because then you can also maybe rely a little or have some support of all the work you did to develop this analytical procedure. It could be some knowledge that could be relevant also for a similar analytical procedure.
I think the next question fits in that pretty well. How does the working group envision balancing ICH flexibility and method procedural instructions with GMP requirements? Now, where did the question disappear to? Oh, there.
Yeah. Well,
specific method SOPs that are executed consistently as written.
Well, we have tried not to go so much into the GMP field, because we always try to be very conscious about what is GMP and what belongs to the dossier. But of course there are gray zones. But when my EU colleagues see something about method transfer, they say, "Oh, that's GMP. We shouldn't write about that at all." So so we should not mix things up and not do things that we are not. But which is not our job. But I can mention that in our discussion group now lately, between the EU assessors, also with the experts from Q12 and Q13 and biological experts and so on, we have now included one GMP inspector. So we try to discuss such aspects where relevant, I think.
We have a lot of questions on Q2 too. There's one from Nadine. Method validation packets are subjected to review and approval by two regulatory groups: assessors at the time of commercial approval and PACs, and inspectors every GMP inspection. Many warning letters are issued by inspectors on changes in validated method years after the product is approved. Is the ICH working group working with GMP laboratory inspectors to assure they agree with the flexibility in analytical procedures?
Yes, I don't hear you well, and it was not easy, but I think the communication of flexibility is a challenge, and I think in our template for centralized application assessment reports, we have an annex 3, which you may have seen, Christoph, where we try to summarize what is the design space and also post-approval change management protocols approved in this application. And also, I think the Q12 tries to say something about cooperation between assessors and inspectors.
Okay, thanks. Maybe last question, time is running. It is great that there will be examples in the Q2 annex, but history teaches that people tend to take these rather as prescription than example. Will there be comments to highlight where examples might be appropriate and where not?
I didn't quite understand the last sentence, but I think we will emphasize in connection with that big table at the end of the guideline that is on only the only examples, and they are not intended to be prescriptive. So that's a very important point. Also communicated by our biological experts. They are really conscious about this, that this should not be taken too strictly.
Okay, thank you.
So I'm getting pushed in the background that we should slowly start transitioning to the roundtables. Shall we take one last question, or?
Okay, let's take one last. How does the working group envision balancing ICH flexibility in method, procedure, instruction? Oh, no, we have the GMP we had already. So sorry,
yeah,
pick another one. So there's a total error as one number expressing both precision and accuracy. Has this been assessed, commented on a statistician in regards to feasibility of such an approach?
Well, I'm no expert on this. We have other experts who know that better, and but I think that the use of total analytical error when it comes to combining accuracy and precision, that's not totally new. I think there is already some knowledge and experience on that.
Right. Okay. So, thank you, Tom.
Thank you.